This week you're going to have another dry lab. This one is on fungi and the algae. And right here on the cover page, front page of your lab handout, that's listed in files right now. And also I think in this week's module, you'll find just some fungus growth. The fungi, they always have these the hyphae, and the massive hyphae, that will be the mycelium right here on the bottom. And then reproductive hyphae, they carry the spores up here. So it's down here and large. You have conidiospores right here. The stalk-like structure, this one here is the conidiophore that carries then the spores. In this case, we have open spores. Also here, this would be a septate hypha. That means we have cross walls right there. You might remember this from the lecture portion on this on fungi. There are septate hyphae and aseptate hyphae. If they have cross walls, then they're septate hyphae. So let's take a look at the terms that you need to know regarding fungi. So the study of the fungi, that is mycology. Myco refers to fungus or fungus-like, ology, the study of. A mycosis will be an infection with a fungus. The thallus is a mass of hyphae, so there's sort of this body of the fungus growth. Mycelium it's kind of similar to the term thallus. A mycelium is sort of all of the hyphae together, and hypha is a singular for this filamentous kind of growth of a fungus. Hyphae would be the plural. They can either have cross walls, and they're called septate hyphae. If they don't have cross walls, then they're aseptate hyphae or cenocytic. A mold, that's a fungus that does not develop fruiting bodies, so it's fungus that just grows. And a sporangium is the how the fungus carries its spores. There are sporangiospores. Those are spores that are carried in a sporangium, in a spore case. A zygospore, that will be the fusion of a plus and minus cell for the type of sexual reproduction that a fungus can do. Conidia, that's another way of carrying spores, so that'd be an open spore case. And a conidiophore, that is what you had on the front page. So conidiophore, this one right here. So the conidia here, open spores right there, and then the conidiophore is the stalk right here. So that's conidiophore. So conidia are the spores of when they're carried in an open spore case, and the conidiophore is the stalk-like structure. Also a fungus is the yeast, and yeast typically those single cells, and they mostly grow by budding. That means a smaller cell comes off of the parent cell, keeps growing, and then it may or may not stay attached as it grows to adult size. So here's some more on the explanations of the terms. And right here, sort of a picture explaining sort of the difference between the thallus and the mycelium. The thallus is sort of the entire body of the fungus, so spanning all of these structures. And here the mycelium is sort of the mass of the hyphae down here. And then here, so this would be vegetative mycelia or vegetative hyphae together. And then up here would be the reproductive hyphae that carry the spores right here. And let's take a look at the lab portion that you're supposed to do for this week. Now, the good news is that in the lab itself, you would be just looking at specimen. And some of them, actually a lot of them, are not super great. So you're not missing much, in other words, if you're doing this online here. First specimen that you were supposed to be looking at is Mycobacterium tuberculosis, and we just had that also in the pathogen group. The name of the disease that Mycobacterium tuberculosis is causing is, of course, tuberculosis. It's transmitted usually through respiratory secretions like coughing, sneezing. You have to have pretty close contact. It's not so easy to transmit tuberculosis, Mycobacterium tuberculosis. But part of body infected is typically the lungs, but it could be also body-wide. Mycobacterium leprae, name of disease, leprosy, Hansen's disease, transmitted by very close contact with an infected person. And take a look at the pathogen sheet there. You have more information. You should just sketch it. For that, in order to sketch that, you should just Google, in this case, Mycobacterium leprae, or previously we had Mycobacterium tuberculosis, and then just do your own sketch on this pathogen. Next one, Actinomyces species. There we have a slide in the lab, and you should take a look at that. You're going to have to Google that and find a nice slide on that and sketch it for yourself and label the sketch. Name of the disease here. There will be actinomycosis, and you should look all of this up. Streptomyces griseus, that one. It's a bacterium that grows in chains, as the name indicates. Strepto means in chains, and therefore looks kind of like filamentous fungus-like growth. You would have gotten a slant culture that you would have been able to look under the dissecting microscope what that growth looks like. Please Google Streptomyces griseus. There are quite a few nice, good images that you will find easily on the internet, and then just kind of make your own drawing and label it. Streptomyces species they are interesting because they're producing antibiotics. Next up, yeast. We have baking yeast usually in the lab, Saccharomyces cerevisiae, and you would have had to dissolve some baking yeast in water and then make a wet mount, take a look, but you can easily find images online and draw your own yeast cells. Candida albicans, that's a pathogen. On our pathogen groups, there we have prepared slides. Please Google that, and also you find plenty of information on the pathogen group, because there we have Candida albicans as a pathogen. Next up, some molds. Those are actually pretty cool to look at in the lab. The molds, we have Rhizopus, that's kind of common, your bread mold. Then we have Penicillium, and we have Aspergillus in the lab. Usually they're grown in slant cultures or in Petri plates, and then you can take a look under the dissecting microscope. But you find nice images. There are some in your textbook as well, and then there's lots of images that you can take a look at on the internet. So please Google these molds, Rhizopus, Penicillium, and Aspergillus. And then ringworm fungus, that one is also in a pathogen group, so you have plenty of information also in the pathogen group. But take a look at that. Next up is Coccidioides immitis, and that one causes coccidiomycosis. Look it up, please. Find some images online when you're Googling this, and make your own drawings. Just a quick sketch. Pneumocystis carinii, we have prepared slides in the lab, so now you have to Google it. Please do that. Plenty of information on the internet. And last, here's lichen, and recall what we had in lecture. Lichen is a combination of a fungus and an alga, so that means we have a symbiosis here of fungus providing a holdfast for the algae cells that then photosynthesize and provide food for both of them. And so that's a nice symbiosis right here. We have some lichen in the lab, but you can easily go out where you find some decaying wood. There's typically, especially if there's some moisture around, you should be able to find some lichen that's growing on, let's say, decaying wood. And you should Google a lichen, and then make your own drawing where you should find sort of this filamentous growth of the fungus, and then embedded into that would be the green algae cells that are having a place to live, and the in the filamentous growth of the fungus, and then they photosynthesize, so the algae cells photosynthesize and provide food for both of them. And here are some sketches. There's some baker's yeast, some sketches that you can use maybe for your own drawings. Here's Candida albicans. That's a dimorphic fungus where you have filamentous growths right here. So these hyphae structures, and then yeast-like growths right there. And then here you have the life cycle of Rhizopus. That's a typical zygomycete. And then here, down here, that would be the sporangium of a spore type of fungus. The stalk is always the sporangiophore. And then here would be another drawing of Rhizopus with the sporangium. Here's some more information. That would be fungus was aseptate hyphae, so the sporangiospores and the zygospores that you can see over here in this panel right here. And then the septate hyphae, here would be conidiospores and the ascospores for sexual reproduction. Again, there's some sketches that you can use for your own drawings. Here, the spore-forming structure of the ascomycete aspergillus. Here you have the conidia. These are open spores, not enclosed in some sort of a structure in a sporangium. And then this conidiophore is the stalk portion right here. Here, kind of interesting, Penicillium notatum. That's the source. That's the mold that grows as penicillin inside. Penicillin means little paintbrush, and you can see how this looks kind of like a little paintbrush right there. But that's the way the conidia, the spores, are being carried in this mold, Penicillium mold. Here would be the cycle of development of Coccidioides immitis. You can take a look at that. And over here, Trichophyton. Then here, the cyst of the Pneumocystis carinii pathogen. And down here, last thing in this lab handout is a sketch of lichen, where you have the filamentous growth from the fungus right all over here. And then embedded into this filamentous growth would be the algae cells. Right over here, you can see it pretty nice. The algae cells that then photosynthesize and provide both themselves and the fungus with nutrition through photosynthesis. All right. Yeah, I know this is not the most exciting lab, but there's really not much difference whether you do this at home or in the lab, just looking at stuff. You can easily Google everything, find images, make your own drawings, and make sure that you study this for our next lab quiz. The next lab quiz will be on this lab plus the pathogen group number 5 that you also find for this week. In addition to this lab here, which is just a dry lab, a paper lab, be sure that you also take a look at the Labster assignments for this week and have fun with this.